Hello students, welcome to Lab 3. In this lab, we're going to talk mostly about visualizations. In combination with our last lab, this lab is probably one of the most important ones in terms of the skills that you'll pick up and apply eventually in your career. First off, it's really important that you understand how to manipulate data, and we made a lot of progress on that last lab. But really beyond that, it's not enough for you to, for example, calculate averages or standard deviations. You need to make a compelling point with the data that you have analyzed. And the easiest way to do that for technical and non-technical audiences is for you to visualize that. So in today's lab, we're going to go over the primary tool that folks use in R to create visualizations. And as I suspect most of you will probably or have already taken a dashboarding course in which you may have learned Tableau, one thing I kind of want to mention is that the underlying concepts and ideas that are used to make visualizations in R are also used to make visualizations in Tableau and many other dashboarding and visualization softwares. And that underlying concept or idea is called the grammar of graphics, hence GG in ggplot. So today we're going to cover just kind of ggplot in general. We went over this a little bit in the first lab where we kind of built up a histogram step by step with data that we had constructed. But since that was kind of a higgledy-piggledy data that I just threw together in 10 seconds, it didn't really have a lot of nice and interesting patterns in it for us to analyze. Instead, today we'll use a little bit deeper of a data set, though still one that's relatively simple, and we can see how many different types of plots we can make from that. So we'll talk about ggplot and why we use ggplot. Then we'll talk about data a little bit, why we bother to have data in the format that we have it. We won't spend too much time on this, but it is important for you to understand why we care so much about what's a column, what's a row, and making sure that our variables and our values are all kind of understandable and take this nice format. After that, we'll talk about aesthetics. And this, along with geomes, these two are probably the most important topics that you're going to want to get your head around from this lab and from the previous lecture. Really, these are the core ideas of how we make visualizations. At the end of the day, we map variables to axes, and we'll get into more detail about what is an axis a little bit later on. So after we talk about what's an aesthetic and what is not an aesthetic or an axis, we'll talk about geomes or shapes. Once you've told ggplot what data should go on what axis, you have a number of different options when it comes to what shape you want to represent that data with. You saw in our lecture that a series of XY coordinates can both be represented by a line or a point, or there are other shapes that you can represent that information with as well. So we'll explore that a little bit. Afterwards, we'll look at facets, which are kind of mini plots, and then we'll look at scales. We talked a little bit about the color scale, but there's really, all of our axes that we match or map in our aesthetic command can be manipulated through scale arguments. And finally, we'll talk a little bit about themes. We won't spend too much time here because in general, kind of focusing on these kind of visual aspects that are kind of not data related isn't the best use of our time right now. And more importantly, ggplot has a number of really nice built-in themes that you generally should probably default to using. So while we're here, we'll also make a couple of different types of plots while we're here. We did one or two in the class up until this point, but I want you to start to stretch your legs a little bit, reach out, look for different types of plots that you think you can make with your data, and then go experiment. So, in this lab, we're going to do 3 different types of plots. We'll do a scatter plot, we'll do a box plot, and we'll do a histogram, and you'll see how those kind of change, how the mapping changes And actually, I think this is the order that we're going to do them in. So terrific. You'll get to see how each of those shapes and the aesthetics matter. So as always, we're going to start off with the tidyverse. We're going to load our tidyverse package, and we're going to use a built-in data set called iris. Let me make this a little bit bigger. So let me go ahead and run that, and we'll take a look at iris. You know, we used this as an example in one of our lectures, so you may already be familiar with it. I believe it was in lecture 1, but today we're going to spend a little bit more time on this. Now, irises are obviously flowers, and there are 4 measurements here for each individual flower that was measured. First, the sepal length, the sepal width. We also have the petal length and the petal width, and the specific species of iris that we have, which are setosa, versicolor, and virginica. So there's only 150 observations here, not a lot, and we've saved that, or we've kind of assigned that to the data object, an object named data. So let's talk about ggplot. ggplot is part of the tidyverse, so you don't need to specifically call ggplot, but why don't we just in case, right? And the actual name of the package is called ggplot2, so you're gonna have to call ggplot2. So let's go ahead and let's just call basic visualization. Let's go ahead and call the ggplot function and see what happens. As it did in our first lab and lecture, when we just run the ggplot function, all that does is tell R, hey, I'm about to make a visualization, and I want you to get this area ready. This is the general kind of plot layout. You can see this kind of white border, the gray area where the data part itself will be drawn, and R is just getting used to that. So great. This is kind of the easy part. This is how all visualizations that we make in this class are going to start. Once we have that, we need to tell ggplot a couple of different things. First off, we need to tell it where to find the data that we're going to be plotting. In this case, we only have one data set. So we're going to say data, and this is the argument. This never changes. We're always going to say data equals, and we're going to say data, right? And this is the name of the object. In fact, why don't I make this a little bit easier to follow? And I'm going to assign the iris dataset to an object called viz for visualization, and I'm going to say viz, right? So let's run this again. Great. No errors. That's always a good sign. Nothing really changed here because we haven't told ggplot yet what it is we're interested in visualizing, which of these variables, right? We can take any combination of these 5 variables, and we haven't told ggplot yet. So all it knows is, hey, I'm going to make a visualization, and it is going to be made with the data in this data set. So terrific, right? So far, we've kind of talked about ggplot, we've talked about data, and in fact, why don't we spend a minute here to think about data a little bit further. One of the things that we're going to cover a little bit later in class is the idea of tidy data, which means that each row is one observation, each column is one variable, and each cell is one value. So for example, what we don't see here is a column called setosa, another column called versicolor, and a third column called virginica. And in each one of these you have a 0 or a 1, whether it's setosa, versicolor, or virginica. That's not tidy data. And we'll talk about tidy data a little bit more. Instead, we have one column because species is the variable that we're interested in. Our observations, each flower varies on this concept, this idea, species. So this is tidy data. We have one variable per column, one flower per row, and we only have one observation per cell, right? This is good formatted data for the visualization process, and we'll leave it here. We'll come back to that a little bit later in the class. Terrific. So now we've told ggplot that we're going to make a plot, first off. We've told it where the data is, and now we need to put some of these variables on axes to understand them. So let's take a look. Why don't we do a histogram first? Why don't we do a histogram? And we've done a histogram before. Basically, all a histogram does is it counts the number of observations or rows that fall into a given bucket. And usually these are put on the x-axis. X is always a good axis to start with. So why don't we start with x? We're going to say on the x-axis, I want you to put sepal length. And we could have really put anything there, but for now, let's just go with sepal length and let's run that. Let's see what we get. Whoops. My bad. Actually, what we need to do is we need to open an aesthetic command. And we need to put it in there. Okay. So there we go. When we run this, let's take a look at what we get. Right? Let's go back and let's look at our sepal length, which is in our first column. And we can generally tell that there's not going to be any values lower than roughly 4.5, 4. And there's not going to be any values greater than 8. Moreover, just like in our first lab, we see that ggplot has labeled our axis for us. We told it that sepal length is going to go on the x-axis. And so it said, okay, I'm going to label this axis this. And I know that however you choose to visualize it, the range of this data is not going to exceed these limits. So I'll just put some nice ticks there for you. I'll put some guides there for you. Very helpful, right? Also notice that this is in an AES or aesthetics function, right? An aesthetic, you can think of it as an axis. So you're used to probably thinking about the X and Y axes, but as we'll see a little bit later on, you can think of all kinds of other qualities of a visualization as axes. For example, color might be an axis because the data varies along some variable that we can map to there. So let's not get too ahead of ourselves. Just remember, if it has to do with the data, it goes inside the AES function. And we'll see what happens when we put things outside of the AES function afterwards. So terrific. We have this. Now, so far we've told it, hey, we want to make a plot. Here's where you find the data. Here's what I want you to put on the x-axis. And now we just need to tell it, here's the shape I want you to use to visualize this data. So we're starting with a one-dimensional, one-axis visualization. We've only told it x, so why don't we make a histogram? I want to see the distribution. Interesting. Very interesting, right? So as kind of noted, none of our values go below 4.5-ish, and none of them go above 8. You can also see that this right here is probably just one observation here, one observation in all of these little guys over here, but then we have, I don't know, let's say like 8 observations that have roughly 6 millimeters of sepal length, or inches. I don't know what the unit is here, right? This is basically how we're interpreting our histogram. There are not many observations over here. There's not too many observations here, and there's a lot of observations somewhere in the middle here, right? As you go to generally either extreme. And you'll look, in fact, why don't I zoom out here a little bit and I'll make this plot super huge. You'll note that it counted the y-axis for us. When we told it, I want you to do a histogram, histograms always have the same y-axis, which is count, or density, but we'll get to that a little bit later, right? So it calculated this for us based on the shape. Don't think about this too hard right now. Think about how sepal length went on the x-axis, right? Okay, that's our kind of basic visualizations. Axes other than x, y. We have this, but when we look at our data, we know that our data, in fact, contains 3 different types of irises, right? Setosa, versicolor, and virginica. And while it is interesting to take a look at the general distribution of sepal length across these flowers, it may be more interesting to take a look at them by species. So if you're recalling last week's lab, you might already be thinking of group by, right? Well, I can calculate, for example, the average sepal length by each group. And indeed, you're right, we can do that, right? Let's think a little bit more visually today. While you can and probably should do that, it's an interesting analytic for you to understand. Why don't we instead, I want to make the same visualization. I want to put sepal length on the x-axis, but this time I want a separate histogram for every species. And the way I'm going to distinguish that is with the color axis. Everything else stays exactly the same. We did a little bit of this in our lecture, so this shouldn't come as too much of a surprise. You saw that once we added an axis, and I want you to think of color as an axis, once we added color, there was a way for ggplot to distinguish between the three different values. It says, oh, I'll just give each of them a different color, right? And we got different colored lines. Here, we'll see something a little bit different. So why don't we run our code and see what we get? Species is not found. Okay, what are the column names? Oops, it has a capital S. Okay, let's take a look here. Interesting, right? One important thing to note, as you'll see, is that color often refers to the boundary, the outline. And instead, what we probably want is fill, the fill aesthetic. Fill will put in the color in the middle here. So why don't we go back and why don't we do that? Let's say fill equals species. Right? And actually, what I really want is, actually I'm going to leave that there. Now, in fact, is a good time to show you what happens when I put arguments outside of the aesthetic command. So these are nice, right? First off, let's take a look at what we've got before we go any further. Then I realize it. We have different histograms. We have different histograms for each of these species. And we can see that, in fact, while they do overlap, Setosa is much smaller in terms of sepal length than, for example, Virginica is. There's no pink basically on the right half of the chart. For higher values of sepal length, we don't see any Setosa. We see fewer versicolor, and we see almost all Virginica here on the right third, the right quarter of the plot. Versicolor tend to be somewhere in the middle, Setosa tend to be a little bit smaller, and Virginica happen to be bigger, right? All we did here was add a variable to an axis. That's it, right? So let's talk about what's an aesthetic and what's not an aesthetic. We've already seen from when we first ran the code with color equals species, that color actually refers to the border outline, right? I don't know about you, but I prefer when my bar charts have a nice distinct little border to them. In this case, I just want a black border around each of the bars. The way to do that is in our shape, I'm going to say color equals black. R is very good at kind of interpreting strings with general colors. You can put like red, blue, green, whatever you want, and it'll kind of understand what you're saying. Notice that unlike fill, I didn't put color inside an aesthetic command. Why didn't I do that? I didn't do that because the color isn't going to differ across my variables. I just want a black border around all of my bars. It's not a variable, it's a constant. So let's run that. And we'll see that, I don't know, I think that looks a little bit nicer, right? Gives it a nice kind of crisp look to it. But notice that it's a constant, right? Constants don't get mapped to axes. So constants go outside of the aesthetic function, Variables go inside the aesthetic function. If you look at your data set and you think to yourself, I want to visualize that, it goes in an aesthetic command. If it's just something that's for the sake of prettiness, it goes outside of the aesthetic command, right? So for example, why don't we, in fact, go back one step, and let's go back to our original, and let's say I don't want to change this by color. If I just put color equals dark red here, and I run this, whoops, actually that's just as good, right? And I run this, you'll see that it made all of the colors dark red, right? Or, whoops, or rather fill dark red. It just made all of them dark red. It didn't separate them by species because I didn't map species to any axis here in the aesthetic command, right? And this is kind of an eyesore, so why don't we get rid of that for now, right? But this really kind of shows you two things. One, any quality of a shape that can change according to the data is an axis. You should think of it as an axis. And there's a difference between what goes inside the aesthetic command and what doesn't. Okay, so we've talked about aesthetics versus non-aesthetics. We've talked about ggplot in general. We've talked about data. Why don't we kind of go on to a box plot real quick? Why don't we consider a different shape? So again, we're going to start with ggplot. We know where our data lives. And if you haven't seen a box plot kind of summarizes what the distribution of your data is. It tells you where the median is from kind of the center line. It shows you the 25th and 75th quantiles. And then the kind of whiskers on your plot show basically the range of your data, depending on whether or not you include outliers or not. So we can look into that a little bit more once we make it. Why don't we create our aesthetic? And I'm going to say x equals sepal, actually, petal length. And why don't we do a geom box plot? Here we go. So compare this visualization to the visualization on lines 7, 8, and 9. In fact, for the sake of continuity, why don't we use the same variable? Same variables, same mapping, same pretty much everything except for the shape. Right? All we did was change the shape. Why don't we run both of these one after another? And when we run the different shape, we can see that we get drastically different visualizations, right? So histograms and box plots are kind of interesting because they do a lot of the summarizing work for you, but all it took to kind of get these vastly different things was just adding a different geom, right? So even once you've decided what variables go on which axes, you need to decide what shape you want to use to visualize that. Again, going back to our example from lecture 1, we saw that we can both make a line, or this most recent lecture, we saw that we can both make a line and a point using the same XY coordinates, and the same thing here, right? So let's go back to our box plot here for a second. As always, it's very interesting to have a box plot of one species, of all of our species together, but I actually want one box plot for each species, and I want to put them on different points on the y-axis. So now we're talking about two spatial axes. Let's go ahead and run that and see what we get. Right. So kind of the original inferences we made from looking at our histogram are basically confirmed here, right? First off, notice that since we defined exactly what goes on our x and y axis, notice that ggplot put those variables on those axes for us. It went, yep, sure, I'll put sepal length on x-axis, I'll put species on the y-axis, and you don't have to think about, well, is this a continuous variable, is it a categorical variable? It doesn't matter. ggplot is smart. It knows what you want better than you know what you want, right? And it did it in a very neat, nice way. Virginica, versicolor, setosa, wonderful. And like I said, kind of our inferences remain the same. Setosa is generally the smallest, versicolor tends to be the next biggest flower, and then Virginica by far is the largest flower, right? But let's say, okay, why don't we bring color into this as well? And let's color our box plot. Again, color generally refers to the border of whatever shape you're doing. And so here we have it, right? And the legend, you'll notice, reflects that. Kind of shows you nice lines there. Notice that we put two, or rather we put the same variable, species, on two axes, y and color. That's not always a good idea, but in this case it kind of makes sense, right? It's not hurting anything. There's not an additional variable that we could have mapped color to. It just kind of makes the three categories a little bit more distinct. Maybe we don't need this legend as we did before since they're already labeled here, but there's nothing stopping you from mapping the same variable to multiple axes. So now we've talked about two spatial axes, x and y. We've talked about color. And we've talked about quite a few geomes already. In fact, why don't we skip ahead a little bit to scales and themes. Now, you'll notice that when we did the ggplot command, but before we added a shape, R and ggplot already knew to make your x-axis a certain width, for example. Kind of the more obvious example is when we say put species on the color axis. I didn't pick these colors, right? There was no point before this lecture where I went and I said, yes, I want pink, green, and light blue. Those seem like reasonable colors to me, right? It picks those colors automatically. And in fact, like these colors are fine, but I may have a greater preference for what colors I want to map those different observations to. So when I want to change things about the scale or the axis, and in this case, the axis could mean the x-axis, the axis could mean the color axis, or it could mean the y-axis, we use scale arguments. So let's say I'm not too huge of a fan of the colors that are used here. All scale arguments start with scale. Then, after an underscore, you tell it what scale you're interested in fiddling with. Clearly, I'm interested in color. And then you need to tell it how you want to fiddle with it. In this case, manual. I'm going to send it some unique values, right? So I'm going to say, what values of color do I want you to use? Well, I have 3 different colors here, so I need to tell ggplot 3 different colors. And I'm going to say the values that I want you to use are, and anytime you put multiple things together, you have to put it in a C function for like concatenate. And I'm going to give it 3 colors. Let's say dark red, navy, and forest green. Let's see what happens. I think those colors are a little bit nicer. It's really a matter of personal taste, right? But as you can see, we're kind of getting in between that step of assigning variables to an axis. We still let ggplot handle all of that fiddly work, but we want to have a little bit more control over what it looks like, right? And to show you that it's not just color that we can change, remember, we can fiddle with pretty much any axis that we want. Why don't we fiddle with the x-axis? So again, anytime we want to fiddle with an axis, we always start with scale. And then we say, which axis do we want to fiddle with? And in this case, we have to say, what type of axis is it? It is a continuous scale, right? Continuous because sepal length can take any value between like 4.5 and 8, basically, right? It's not like an integer like 1, 2, 3. It's not categorical like these three, right? It's continuous. A flower can have any length between here and there, so it's continuous. Maybe I want my plot to, in fact, show a much larger area. Maybe I want the limits of my plot to start at 2 and go to 10. So maybe I'll say limits equals, again, C, because we're giving it two things, the starting point and the ending point. And let's run and see what we get. Right. So this is kind of counterproductive in the sense that ggplot actually usually picks pretty reasonable values, right? But in case I want to zoom in on a specific spot, in case I want to zoom out, this is not a bad idea. It's something that we can do. I'm going to comment this out for a second, and we'll just kind of go back to the general idea of, hey, you have scales and you can use them. So anytime you I would say color is probably color and fill are usually the scales that you'll find yourself fiddling around with the most, right? So we've talked about scales. There are other ways that we can fiddle with scales as well. Let's say that, I'm not so good actually at picking out different colors. I just want to use a pre-made nice color palette. There are actually many pre-made color palettes in ggplot. So let's say scale, again, color. I'm going to say Viridis. So Viridis is a color palette that is very popular. And this is a discrete variable, right? Categorical. There are discrete quantities here. There's 3 of them. And all I have to do is say that, let's comment this out for a second. Let's run this and see what we get, right? So it picks these colors from the Viridis color palette, yellow, teal, and purple, if you're into that sort of thing. And, these have some nice qualities to them. Like, for example, they're colorblind friendly, other things like that. If you ever work in a public environment doing research of that nature, it might be required for accessibility reasons that you use colorblind palettes and friendly palettes and things like that. So, if you don't want to think about what values you want to make your visual, these scale arguments are very helpful. Let's go back to our green, red, blue example because I think that looks pretty nice. Okay, last thing before we kind of move on to facets, and let's say we've done box plots now and scatter plots, is the theme. You'll notice that there are kind of a lot of visual aspects of this plot that are not directly associated with the data. Kind of the most glaring thing is that the background, the standard background of a ggplot plot is this gray color, light gray color, with white grid lines. It's very distinctive. If you just kind of use this standard, people will know exactly what you used to create this visualization, but I don't think it's the nicest looking thing in the world. And what really bothers me, kind of what we were talking about, I don't think we need this legend here. It takes up a lot of space. It's kind of redundant since they're already on the y-axis. So I want to get rid of that. So for everything that is on your visualization, but is not related to the data, we use the theme argument. So theme. I'm going to open up a theme argument. And you can see that there's so many different arguments that you can use here. You'll kind of have to noodle around to find the specific parts of the plot that you're interested in. And we're going to say legend position equals none. That is not a very intuitive way to do that, I know, but it's just kind of some of these things that you're going to have to figure out as you do your assignment and stuff like that, or as you fiddle with your visualizations. And as you might expect, when we run this, it'll just get rid of the legend, and we didn't really lose any important information because, again, our species colors are already here on the y-axis. Likewise, if I want to make a more general change to this plot in terms of the visual aesthetics of it that are not data related, I can use one of the built-in themes, just as there are built-in color palettes. Now, I like theme minimal, and if you run this, it kind of applies this very basic, standardized aesthetic, right? It took away the gray background. It made the grid lines light gray. Now everything's kind of white. The colors are all kind of a little bit more distinct on the axes. Things like that, right? Things where if graphic design is not your passion, you should default to this. One thing to keep in mind is that if you want to do any specific fiddling like we did, like getting rid of the axis, you're going to want to put that after your pre-made theme because you're kind of updating that, right? So there we go, right? I think this looks quite nice. I think this is a nice visualization. So let's cross that off our list. For non-data related aesthetic things, you're going to look in your themes. If you want to fiddle with the axes, you're going to look at your scales. And one of the last things that we should look at is probably facets. So while we're at it, why don't we look at our scatter plot? Why don't we create a scatter plot rather? We'll pull up our data one last time, take a look at it, and by now we're getting pretty good, I think, at making a visualization. We always start with ggplot data equals viz, or whatever your data set is called. We're always going to open up an aesthetic command. And we know from experience that scatter plots require two aesthetics. First, the x-axis. When we do sepal length, and a y-axis. Why don't we do petal length? And then, first off, why don't we run that to make sure that, yep, it recognized it, it put those variables on the different axes, and it got the range relatively correctly, right? Now, why don't we just add, oops, not a histogram, geom point. Ta-da, right, there we go, there's our points right there. Just like that, in 4 lines of code, we got a nice scatter plot. And you know what, we kind of have the same problem that we did last time, so why don't we go ahead and add species to the color axis. Okay. I believe this is the plot that we made in the lecture, but it's good to kind of take some time and go through it again. One of the ways that we can separate data is by facets. And again, as in the lecture, facets are kind of miniature plots, multiple miniature plots. When we want to make a facet, a good argument to use is facet wrap. Facet wrap. This tells ggplot, hey, I want you to make multiple plots based on the name of the variable. In fact, I'm interested to see. Nope. Okay. So when you do facets, you have to put the name of the variable in your data set in quotes. So let's go ahead and run that and see what we get. Okay. Why don't we do some things to clean it up here, because this is kind of difficult to look at, I would say. So I'm going to say theme minimal, and I'm going to get rid of the legend again because it's duplicated here on the facet label. So let's go ahead and do that, right? We're going to make it pretty. We're going to get rid of the legend, and let's see what we get. Okay. Let's go ahead and zoom that out a little bit and see what we got. Again, the strength of ggplot is the concise and consistent language that you can use to create complex plots like this. Yes, having all of these observations on one plot can be helpful. You can see the overlap a little bit better. But putting them separately also tells us something. First off, like how cohesive each of these species generally is. You'll get one little outlier like that, for example, but generally speaking, versicolor, virginica, and setosa tend to be very similar to each other, and they tend to be dissimilar from each other. And we learned that because of the choices that we made when we did our visualization, right? Always remember that even after you make your visualization, you have to interpret what you're seeing. What is the point that I want to take away from here? There can be many different points, right? But your point in general should be relatively obvious. And in this case, facets really helped us to get there. Virginica does not vary across the entire range of petal length, right? Petal length seems to be a very discriminating feature, discriminating in the sense that it helps us tell these different iris types apart from each other. And again, facets helped us do that. So terrific. We've got scatter plots done. We've done facets. We've done scales. So although this is not an especially long lab that we've done, you should really be coming away from this lab with a couple of core lessons that you take with you going forward. First off, I cannot emphasize enough how important it is for you to get good at visualization. I'm speaking on a bit of a personal note here, but the one skill that has helped me throughout the course of my career consistently get well-paying jobs is my ability to create visualizations. As a data analyst or data scientist or whatever career you choose, it's not enough for you to be able to do really complex analytics, right? It's not enough for you to be the smartest smarty pants in the world. You need to be able to communicate your findings to other people. And in fact, most of the time, very simple analytics paired with powerful, obvious visualizations will get your point across so much better than telling someone, well, it's statistically significant, right? Now, rest assured, pay attention to your statistics courses because those are very helpful as well. But again, if you can make a point so obvious that even a child can understand it looking at your plot, then you've kind of won in a way that's, complex statistics cannot get you there, right? So I really want you, after you've worked on this lab, hopefully you've been following along in your own code, but what I really want you to do is to start playing with visualizations. Start looking at different types of geomes. Start looking at what types of visualizations are out there, and can I implement them in the data sets that I'm interested in? And when I do, what do I learn? Right? Because as easy as it is to look at means and standard deviations, it's much, much easier to just look at a scatter plot, for example. If your pattern is apparent on a scatter plot, you don't need to look at the mean, or rather it gives you a greater context, right? You're never too good just to make a scatter plot. You're never too cool just to make a histogram. So today we covered maybe not too much, but really important stuff. First off, we talked about ggplot. We've gone through by now several iterations of how you build up a plot. Then we talked a little bit about what the structure of your data should look like. We also discussed what an aesthetic or an axis is and what is not an aesthetic, and this comes down to the difference between what is a variable and what is a constant. Then we talked about geomes. What's a shape? And how does that shape influence what visualization we're making? We showed the same aesthetic mapping across two different shapes, which was interesting. We did facets, kind of most recently. We showed little miniature plots. And we also did scales and themes. And these showed us, one, how do you, with scales, get in between the mapping and the output, right? In terms of I want to pick the colors that get mapped to these values, or I want the x-axis to actually be a little wider than the default. And in theme, theme just boils down to how do I make this visualization look pretty, which, by the way, is very important. So it is important in your career. Pretty visualizations will get you a long way. Lastly, we looked at generally three types of visualizations. We did a scatter plot most recently, we did a box plot, and we also did a histogram. We've only covered these three. Again, please explore as much as you can. So I think we've covered a fair bit today. Go out, explore, work on your homework, start thinking about your final assignment, and I hope you have a wonderful day.